Hello everybody, today we're going to talk about gene regulation, and we're also going to talk a little bit about some of the stuff that I talked about in the last video, where it was about protein synthesis. And I'm going to talk about a few facts that I talked about that might be misleading. So let's get started. First, we start with the misleading fact. Well, it kind of makes sense, but when I was explaining transcription, and so if this is a gene, I talked about how there is a sigma factor sitting on the DNA, which is the hardware for the information gene, and a RNA polymerase flies by, and the sigma factor catches the polymerase and puts it inside of the two layer of genes. I mean DNA, the two opposite sides of it. Well, it's a double helix, so you can. It looks kind of like a ladder, and there's kind of links in between. I said the RNA polymerase is put between the two sides. Before I draw the gene, let's draw the polymerase first. Well, a RNA polymerase is made out of 6 parts. There's a part called beta, part called beta prime, and there is a omega, 2 alphas, and one sigma. And so, well, if you look here, I talked about last time a thing called sigma factor, and I read a bit more and found out that RNA polymerase, there is a sigma factor kind of attached to it, but this time the sigma factor, the subunit sigma, is kind of loosely attached to the polymerase, so it has a low affinity for the whole thing, so it can be easily taken off. So the RNA polymerase kind of finds a piece of DNA to sit on, and it's now kind of like this with the sigma right here. And the polymerase does not know where to be. It does not specifically sit on a certain part of the gene, and it does not know where to go. And the sigma factor guides the polymerase and guides the polymerase where to sit on and start transcripting. So, and this is, let's say, from this point is the part of the gene where that has to be transcripted. Let's call this position +1. And now a bit backwards, there is a part -10, and there is a -35. On these two, there are special codes for the sigma factor to bind on. So there are kind of codes made out of the nucleic acid bases. Oh, and last time when I talked about bases, the A, G, C, T, U, they are more like they are nucleic acid bases. They are nucleic acids, and before that, RNA polymerase always grows the RNA following the 5 to 3 direction. So the 5 end to the 3 end. I don't know if you remember, but a part of DNA kind of looks like this. The black things are carbon, and the white things are oxygen. This is a minuscule part of DNA, and this is the nuclear acid base. I don't know if anyone calls it NAB, but this is the base. And now we count from here the carbon. 1, 2, 3, 4, 5, and this is the 3 end, and this is the 5 end. So the RNA polymerase always makes the RNA following this direction. So because RNA is kind of put right next to the DNA to catalyze it, these thingies are put right next to the DNA to catalyze it, to make it so the polymerase follows the 3 to 5 direction of the DNA to make RNA go this direction. So on the -35 part of the gene, there is a sequence of bases going sequence of nucleic acid bases going T, T, G, A, C, A, and one going T, A, T, A, A, T. And of course there's one beneath it going A, C, T, G, T, and A, T, A, T, T, A. Well, it's kind of like this, and the main part of the RNA polymerase, the upper part, does not know where to put itself on. Well, I kind of drew the polymerase like this, but it's made up of multiple proteins. As you see, the subunits are separate proteins. This is a complex protein made out of multiple enzymes, and so it's kind of a big thingy. So I'll draw it kind of like this. It needs to be in this kind of setup, right on the 35 and -35 end and the -10 end, and if it sits right there, it can start transcripting the one end. Maybe it needs to be a little bit bigger, so it can touch the one end and starts transcripting. So the sigma subunit guides the polymerase to sit here. So and this part, these sequences are from E. coli. Other cells might be different. Oh, and this is all for prokaryote cells. Prokaryote cells have the special sequences for transcripting. Prokaryotes do that, and this is a sequence for E. coli, and now the sigma factor guides it and falls off when the polymerase is in place. The sigma factor falls off, and as it falls off, the polymerase starts transcripting. And near the DNA, there are a lot of NTPs around. What is NTP? NTP is nucleic acid triphosphates. So basically, it's the AGCTs, or in this case ATGC, and U is also here around. So this is the base, and there is 3 phosphates, right? 3 phosphates right next to it. It's stuck on there. So if you remember my series about biology, it was called Tree. I explained about how energy is made in cells. You should remember ATPs, which are adenosine triphosphates. Adenosine triphosphate is a molecule that is used as energy sources. It is the currency of energy for cells. ATP transports energy, and how energy is stored is with the triphosphates. These molecules are phosphates, and there are 3 of them, so triphosphate, TP. And the phosphates don't like each other. They don't want to stick next to each other, but they're stuck next to each other, so they try their best to get out of that situation. So when a ATP releases energy, how ATP releases energy is it kind of touches something that needs energy with the phosphate end. So and because the phosphates don't like each other, it kind of becomes a big kick, and that gives energy. And the ATP becomes ADP, which is adenosine diphosphate. Only has 2 phosphates, less unstable. It's more stable, and it can't give energy to enzymes and stuff. So this NTP, this is the base. The NTP is sucked up the beta primes, the beta prime and the beta, the subunits, and it is sucked up there. And let's draw another one here. This time with the, so the things go up here, and also the sigma factor kind of, well, it was sort of right. Some part it was this is kind of right. Some part of the RNA polymerase is put inside of the gene, inside of the DNA, so in the slit of the 2 sites, and starts catalyzing the RNA. So some part in here is inside the DNA like this, and RNA is made here because and the bases are sucked up and it is put together. And also, I said this is a triphosphate, and I also said that adenosine triphosphates derive energy. They bring energy using the phosphate, the 3 phosphates. When a NTP goes in, the 2 two phosphates come off and give energy. It releases energy to put itself onto the growing RNA. So why it has three phosphates is because there needs to be energy to stick RNA thingies together. So basically these come off, release energy, using that energy puts themselves together and becomes RNA. And the rest is explained in my last video. So actually I think this is about all for the explaining thing about last episode, what were left out. So now let's get on with the main part of today's episode. So I said today we're gonna talk about gene regulation. So what is gene regulation? Gene regulation is exactly what its name says, regulation of the gene. So we talked about protein synthesis, but a gene can't be continuously making the same protein at a fast rate over and over again. The gene might not need the protein, and in some cases need the protein, in some cases not need the protein, and sometimes maybe having the protein could be dangerous. So genes need to be regulated. If it's not a very simple creature, some prokaryotes do not regulate genes, do not do anything to stop the protein synthesis. But well, most even prokaryotes do the regulation of the genes. So now we have a gene here, and this is a RNA polymerase. It wants to move this way and start making the RNA, but there is another protein binding over here. It is blocking the polymerase from making the RNA, and sometimes it unbinds and lets the polymerase do what it does. So that is how it works sometimes, and I'll give you an example of E. coli. I said the sequence of bases that start the polymerizing was also from E. coli, and I think E. coli is used in a lot of experiments about biology. E. coli is used a lot because maybe because it's easy to get. If you study biology, I think you're gonna hear a lot about E. coli. So in there is a thing called LAC operation in E. coli. What that is it is basically an operation for breaking down lactose. So here's a E. coli. I don't know if it looks kind of like that, and here is a lactose. Lactose is sugar and can give you energy. So does the E. coli want the lactose? Yes, of course it does. It gives you energy. It's good for you. So inside the E. coli, there is a gene. Actually, we're going to look at 3 genes. It's in the order of Z, Y, and then A. And let's say here is the polymerase, and there's the sigma factor here binding the polymerase to the right spot. But it wants to go and make the RNA for all of these, but there is a protein blocking it. And what these genes does is, well, what the LAC gene does is it produces a protein that actually breaks down the lactose into glucose, so the E. coli can digest it and turn it into energy. So LAC breaks down the lactose. LAC brings in the lactose, because if there's lactose around the E. coli would want the lactose. And it opens the membrane of the E. coli and brings in the lactose and closes it and brings in more lactose. The LAC protein brings in the lactose, and LAC-A, we don't know what LAC-A does. But it still is in the LAC operation. So a lactose flies by. Well, but this protein is right now blocking the polymerase from moving. The sigma factor has not yet fallen off because it can't do its job. But if a lactose flies by, this protein binds onto the lactose. So if this is a gene, and so imagine a protein that looks kinda it kinda looks like this, but only the handle end. So it's both the handle ends. It is made out of two handle ends that can bind to stuff. So one handle is binding onto here, and the other handle is wide open, ready to bind to something else. This is kind of just a simple analogy to if one binds, the other is open. So here is a lactose. So then it binds onto the lactose, and this part opens. So then it is a free gene without any proteins bound to it. Let's draw something over here. The protein binding onto the lactose. So when the lactose kind of passes by, the protein binds to the lactose instead of the gene. So the gene is free from the binding, and now the polymerase can be polymerazing. Now the polymerase can do its job of making the RNA. Yeah, and LAC-Y, the gene for the LAC-Y protein, brings in more lactose if lactose is still around, and it keeps on the process. And so the E. coli can use the lactose as energy. And yeah, this is kind of how it works. But there is a kind of a exception. When glucose is around, the simplest sort of sugar, and because it's the simplest, it can be kind of made right into energy without being processed like lactose. And lactose has to be processed, which needs energy. Lactose uses a little bit of energy to be processed, so it is not as efficient. But glucose is the most efficient energy sources out there for the E. coli, and well, basically for all living things on Earth. So if there is also glucose around also, the protein does not bind to the lactose. Well, it might bind, but it still binds onto the gene and does not let it pass. So in this way, the E. coli always processes the glucose first. That way, the E. coli uses glucose as its primary resource, and it really likes glucose. It always eats glucose first. And this is basically how gene regulation works. And also there's another sort of regulation for genes, and how another way to regulate genes, and it is if we have a gene here. Let's say there's a RNA polymerase trying to make RNA. The sigma factor can bind over here, and also over here, also over here. And this part, let's say it is a part where a RNA polymerase could start making the RNA, but in some places it is made so that the sigma factor cannot bind. So, let's say something happens, like temperature rises. Some part of the gene, a protein unbinds and produces a protein called sigma 32. A sigma factor is, well, the most common sigma factor is sigma 70, and because sigma 70 is the most common, sigma was given to this protein. The reason for its name is because this sigma 70 has a weight of 70,000 daltons. Well, sigma was the Greek alphabet, meaning that was used for the number 70. So sigma was given to this protein, to the name of this protein. And so, but sigma 32 is 32,000 daltons, I'm pretty sure. And this sigma 32 is made when the temperature gets kind of hotter. And this sigma 32 makes the polymerase able to bind somewhere like these, so it can make stuff that is fit for higher temperatures, proteins that are needed for higher temperatures. It kind of works like this. And also, gene regulation is highly needed for us. It is basically used in almost all of the species on Earth. As I said before, some don't use regulations, but if we as humans, as Homo sapienses, stop regulating the genes, we will pretty much die automatically. And regulation is that much important. And also, there is a gene called Homeobox gene. This is a very important gene. Homeobox genes, or Hox genes for short. These are the genes that regulate the whole 46 chromosomes that you have in your body, you have in your each cell. Homeobox genes are the genes that regulate every gene. And so it is basically a gene encoding what should be regulated. So when a baby is growing in a womb, it starts from one single cell. The cell reproduces. The cell becomes more and more, and well, there needs to be organs, and some needs to be. There needs to be organs, like some needs to be lungs, some needs to be the liver, some the heart, and the brain, and so on, muscles, and blah blah. How those diverse cells arise from one single cell is, we can thank the Homeobox gene for that. The Hox gene kind of regulates everything and chooses what cell becomes what by regulating what protein should be synthesized, what protein should be made from what chromosomes should be regulated and shut down. These stuff makes the diversity of multicellular beings possible. And also, some regulated genes regulate some other genes after being regulated. So if some gene is being freed from being shut down, that might produce a protein that opens up another gene and makes it more makes a more complex part of your body and stuff. I think I said everything I wanted to explain today, and any questions or something that might be wrong, leave it in the comment section, and bye.